this is my topic, and I want to thank you for inviting me and giving me the honor to show our data. I want to start with this slide that you all know, basically. This shows lethality of people against COVID infection, depending on the age group. And you see that there is this particular sensitivity of elderly people older than 80. And that's why they were put into the first priority level to be vaccinated with BioNTech vaccine, as you know. And so we relatively early asked the question, can we indeed find, depending on this vaccination, a proper immune response exactly in this population of people? This means this group of people. These are three groups from Marburg, virology, microbiology, and immunology. And we joined together with a group from the DRFZ, Hendrik May, with whom we have now a very fruitful, very long-standing collaboration, and you will see that. I'm afraid much of what I now am going to show is a bit of a repeat. I excuse you for that. Some of the speakers have mentioned this before. So if we have a proper immune response, we have to start with the innate immune response, so macrophages and dendritic cells. And as Gunther has already told us, they have these receptors, toll-like receptors and other type of receptors, and they react with DNA, RNA, with constantly present things on the surface of bacteria. And by reacting, they produce a kind of a danger atmosphere, which then the microenvironment is kind of a startup where the adaptive immune response joins in. And we have the B cells, and we have heard about this, that at a later stage as plasma cells, they produce antibodies. And we have the T cells, which help the B cells, but also are cytotoxic by themselves and can kill virus-infected cells. And in the report, I will concentrate on our analysis on T cells and antibodies. I will not show any data on innate immune responses produced by Stefan Bauer. So as for the antibodies, we have heard about that. We have up to 5 × 10⁻13 different specificities. They recognize their antigen in a 3-dimensional way. They bind the virus, for instance, in serum. They take it away to a phagocytosing cell and neutralize it. And it's relatively trivial and easy to measure this. And you are all familiar with these BAU units that you get in any laboratory. And this is by and large taken as a means to measure the height of the immune response. But of course, this only shows you one part, and it's the part that is usually presented because it's easier to be measured. This is the T cells, and their specificity are even higher, 10⁻18 different specificities. And as you may know, they don't react with a really 3-dimensional way of the foreign antigen. Rather, this is a part of a protein which was produced inside of the cell and which was cut into pieces and which was inside of the cell hooked into the groove of these MHC molecules, which are the transplantation antigens, which you all know. If you want to be transplanted with a kidney, you have to be typed for that because all of us have a known collection of these things. And then this is brought up to the surface and it is recognized by the T cell. And each T cell has only one type of receptor, but has obviously many, many copies of this receptor. So if this T cell is reactive, it does many things, among which we use 2 different things. And this is upregulation of this molecule. And Petra Bacher has shown that. And the molecule has actually many years ago been introduced by Andreas Thiel and Alex Sheffold, because you can use this to monitor antigen-reactive T cells. And the other molecule is interferon, which is an antiviral molecule. And you can use dyes to stain these 2 molecules, and that's what we did. And this is a typical picture of such an analysis. Each dot is one cell, CD40-like in this direction, interferon this direction. And you see these are the interesting cells, and they appear after overnight culture of PBMCs, which contain the T cells, in the presence of the virus. They don't, this is the first important thing, they don't show up if they are not cultured with the virus. The second thing is there are more of these cells depending on the state of vaccination. After second vaccination, you find them here, compared to before vaccination. And the third thing is this test has a remarkable sensitivity, 0.02%, which is far, far beyond what you can see if you measure, for instance, interferon in the supernatant cells. So this is the basis of our analysis. And this is our group. We have 51 vaccinees, 80 years old. We have collected them first in a vaccination center because we wanted to select people that are healthy. We did not want to collect people that are staying in an elderly home because that would probably be a selection for people that are not so fit anymore. And we wanted to compare this to really fit young people. But I can assure you in the follow-up, this is a not so trivial task to have this group together in the countryside around Marburg to collect samples everywhere and anywhere. And it was not an easy task to keep them busy. So we compared this with people that were actually in an elderly home and the same age, but who had recovered from COVID-19. And all of the vaccinees had received 2 doses with the BioNTech vaccine 3 weeks apart. So we took PBMCs, we stimulated them in vitro overnight, we fixed them, we stained them, and we sent them to Berlin for analysis. Or we collected serum and analyzed the serum by routine methods. And this is the first result, and it shows you the result for antibodies. And it shows you first that you get an increase by the first, but more so by the second vaccination. You do see this increase in old people, but also in young people. You see that young people react with a higher antibody level. And you see that the young people react in a much more homogeneous way. In the elderly, you have this cloud much more open, and you have apparently some people who react very, very poorly, and some do not react at all. And if you compare it to the recovered, you have the same heterogeneity, and overall, the amount of antibodies are somewhere between the first and second vaccination level. You go for the T cells, you find more or less the same thing. Increase per vaccination, increase in elderly, but also in young. Increase in young much more than in elderly. More homogeneity in the young. And some in the elderly that apparently don't seem to react. Now, the immediate question is, are these that don't react with the T cells, are those the same that don't react with the B cells and antibodies? So we made a correlation analysis. And this is what you see, and you see this very striking homogeneity in the young again. And you see this open curve, this open cloud in the old. And you see these red marked people that don't seem to react neither with T cells nor with antibodies. And of course, we cannot prove that, but of course, we would consider them to be non-protected by this vaccination. So we immediately went on, and long before the third boost was integrated, we boosted them a third time and showed what happened. And the result was remarkable. By this third vaccine, we could get the majority of these primary non-responders to respond. And this was true for antibodies, and this was also true for T cells. There is one individual, and this is this one, and this is an 84-year-old man who did not react by the third vaccination as well. And meanwhile, he did not react to a fourth vaccination. And he has received, in addition, 2 doses of Johnson vaccine meanwhile. And now, after this actually 6th vaccination, he has reacted in a proper way. So we don't know whether this is due to the different way of the vaccine or whether this is now the 6th dose. I can tell you that he has one brother who with the same vaccination has more than 2,000 units of antibodies. And he has another brother who reacts just like himself. And obviously we are in the process of sequencing the genes of these 3 people. What about, I should mention that these non-responders, they are just non-responders for COVID and for the COVID vaccine, because they are in the average of everybody concerning measles. All of these people apparently had measles infection before they are old. So this is what remains as antibodies for those people. And if you use a mitogen for the T cells, you find that the SAB reactivity is also very much in the average of everybody. So what about the majority of people, those that are not non-responders? If you give them the 3rd vaccine, and what happens after that? And this is now unpublished data. This shows you first the antibodies. And this is what I showed you before. And this is now only elderly people. After 5 months, 5 months after the 2nd peak vaccination, the peak response, you see this waning, and this waning has now been addressed by many speakers before. If you revaccinate them in the booster 3rd time, you get nice information, you get a beautiful boost, and the antibody levels, as Andreas has already mentioned, are higher than what you get after the 2nd boost. And if you further observe this, and these are now values that are 2 weeks old, you see the waning. The waning apparently is much smaller. So I consider that in principle as good news. We may keep antibodies for a longer period of time due to this 3rd vaccination. However, if you go for the results, the early results now from Israel, it looks as if this 4th vaccination really has no impact on protection against the Omicron variant. And I should say what you observe in this waning period here, this is not just going down to enough remaining memory antibodies, because in this period we have now elderly homes in which we find people that are vaccinated twice, and they have breakthrough infections, and they have severe courses in these breakthrough infections, and 25% of those people die. And so it's not so trivial whether we can save them from dying in this phase as well, but I would hope so. But since Omicron is not really protected, we need the T cells, and for the T cells, I have less good news. They wane, like the antibodies, between peak 2nd and then before 3rd vaccination. But the gain that we receive by the 3rd vaccination is not higher than what you get after the 2nd, and there may be even the impression that it's actually lower. And we don't show the waning so far, but the waning goes down in a speed that is very much like the speed after the 2nd vaccination. So since we need the T cells, and concerning an overall suggestion, should we be vaccinated again, I would tend to say yes. What the speaker also says, that we probably need a 4th vaccination in these elderly people to maybe keep at least this level of reactive T cells up. And I want to mention that we have one patient actually who suffered from hairy cell leukemia, and who was treated to get rid of any B cells, and he received COVID infection while he had no B cells. And we met him half a year later, and he was healthy, and he had no leukemia, and he had no virus, and he had survived. And he still had no antibodies, absolutely no antibodies. So I'm quite sure that we need the T cells, and maybe really only the T cells, to survive the infection, whereas the antibodies may protect us from getting the virus at all. So I think this is the important part, and maybe this makes sure that we require a 4th vaccination. One final word for quality of the T cells. We still didn't measure memory T cell markers, but we have measured the interferon production per cell as a marker of maturity, and it's exactly the same. So between here and here, we see maturation, we see more interferon per cell, but between here and here, the amounts of interferon are the same, indicating that also with respect to the intracellular interferon as an effective marker, we see no further maturation between second and third vaccination. So this is my summary. Immune responses in elderly are lower as compared to young individuals. 90% of elderly respond nicely. However, we have primary non-responders, but in most of them, this is not fateful, but can be overcome by a third vaccine. We observe a considerable waning of the immune response after the second, and less so, obviously, after the third dose. And the antibody, but not the T-cell response, are triggered by and are higher by a third than after a second vaccination. I want to thank you for your attention, and I also want to particularly thank the vaccination center in Marburg and especially its executive head, Carsten Oeder, for protecting us throughout this study. And thank you for your attention.
Yes, thanks a lot for the very nice presentation, Michael. I'm looking at the chat and I can see two questions. The first is from Andreas Radbruch. Do you want to ask a question?
Yeah, Michael, it's a very simple question. So this sounds like adaptive immunodeficiency in those 5 people who responded late and the one that didn't respond, that responded very late, actually. So the obvious question is whether you measured antibodies against interferon, type 1 interferons, because type 1 interferons are also known notoriously as differentiation factors for plasma cells. So it would actually, depletion of the type 1 signaling would ablate actually antibody production.
We did not measure the level of autoantibodies, but we did look at all these patients for their comorbidities. And this 84-year-old man is very healthy. He's a sportive man. He has nothing that you can find. One of the 5 was treated with methotrexate for rheumatoid arthritis, but there was nothing special about these 5 people in comparison to all the other people. We monitored drug medication, we monitored all types of other diseases. There was nothing special about them.
Yeah, but the measles is a bit unfair as a comparator because that's really established in young age. Yeah, so it just means that the plasma cells who these people have since many years are still working. But the question is whether other viral infections like influenza also whether they have a special comorbidity for viral infections.
Nothing.
Susceptibility.
We have squeezed them as medical doctors. We have followed their history as tight as we could. There was no difference to be observed.
Okay, thank you.
Lots of more questions. So next one is Thomas Dörner, who asks importantly about the comorbidities.
But I think, Michael, thanks very much. You somehow answered this already with the patient with RA methotrexate. It's pretty unusual that people above the age of 80 have not seen a doctor or suffering from any comorbidities. But
well, no. This is what I didn't want to say that. I wanted to say that the comorbidities in these young people were spread around by chance.
Okay.
You could not find any particular hint on anybody that would react or would not react. Yeah? I have 2 people that reacted very nicely, and they are very sportive. That I can say, but this I think is not a p-value.
Yeah, but you should patent the rituximab vaccination strategy for getting rid of hairy cell leukemia.
Okay, Christina asked about the HLA haplotype.
No idea.
That's exactly the point to many T-cell assays. Is there any idea whether the haplotype plays a role? Because you see this variation, and it could be, but probably there are too many peptides so that it's sort of equalizing then the haplotype question. I don't know what your idea is, and also Alex and Petra, of course.
As you know, the set of peptides that you buy, they are overlapping. So they should in principle collect everybody. I need some hope into these 3 siblings. So the 2 of which don't respond at all, and the one who responds nicely. And actually, they even have a sister who answers in a correct way. So we have 2 and 2. So I would hope that if it is the HLA, we would make a nice discovery. But I have no data so far.
Can I just comment? Because in Kiel, people looked very much for HLA association with anything, but there is nothing.
Interesting. Okay, Dirk Görlich, please.
Yeah, very impressive. I mean, the take-home message is that one should actually try everything to identify the non-responders and give those specifically another shot. Is this practical within our medical system?
This is a very good question. And in addition, I must tell you that among those, apart from those that don't react at all, we also have people that have high loads of antibodies but no T cells, and the other way around. So if you would screen only for antibodies, which would be relatively easy, you would still miss out people that have a tremendous T cell response. And if I mention this patient again, who had the disease, he was like crazy to be vaccinated. He said, I want to get my Persilschein, yeah? I want to go everywhere in the cinema. I need to be vaccinated. So we didn't intend to vaccinate him because he had no B cells. And we said it doesn't make sense. So we waited for about 8 months until the B cells came back. And then we vaccinated him. And he had received so much of side effects. And probably because he has just 5 times as many T cells. So if you would select, and you would select only for antibodies, you might miss out people that have no antibodies but have decent amounts of T cells, and you would boost them and they would really have side effects. So it's tough to be answered. I would say that if you select only for those that have no antibodies, you maybe overall do a good thing. So if you would skip out the result on T cells, it's in our collection, it's relatively rare that you have only T cell responses. And to select for B cells and measure BAU units, I think that in principle would be doable if the ministry would give it enough money. So that's a long answer. So in short, I would say yes. For me, it would make sense to measure antibodies for everybody.
Okay, the last question by Alexander with a request for a short answer.
Even the question is long. But just I want one particular point. You said the quality in the elderly, the quality of the T cells is not different. This is different to our results. I don't know sort of quality in terms of interferon gamma production. So for example, what we clearly saw is that they make definitely less IL-21, and especially they have low affinity. So our interpretation would be that in a certain age group, you really will have difficulties to generate good T cell response simply because they have not the right TCR affinity available. So my question
Alex, I may interrupt because I may have been misunderstood. Sorry for that. I did not intend to say that old and young don't differ with respect to quality. What I wanted to say is that within the old, we don't see further maturation of quality between second and third vaccination. And we do, I cannot show you the slide again, but actually here the recovered are much more different. The recovered have a higher quality of the T cells. So what I wanted to say is that by the third vaccination, we are not able to approach more the quality of those seen in the recovered. So something is missing out to get the T cells this final kick towards what they experienced due to true disease.